Welcome to IIOCS.com Food Tech Club. Today's topic is role of fermentation and chemical preservatives in food preservation. Let us see first about the role of fermentation in food preservation. The term fermentation is derived from the Latin verb fervere, that is to boil, thus describing the appearance of the action of yeast on the extracts of fruit or malted grains. The boiling appearance is due to the production of carbon dioxide bubbles caused by the anaerobic catabolism of the sugar present in the extract. However, fermentation has become to have with different meanings to biochemists and to industrial microbiologists. So that we will look in further slides. Its biochemical meaning relates to the generation of energy by the catabolism of organic compounds, whereas its meaning in industrial microbiology tends to be much broader. So what do we call the study of fermentation, the science of fermentation? It is nothing but zymology. The study of the science of fermentation is zymology. Fermentation processes are believed to have been developed in order to preserve fruits and vegetables for times of scarcity by preserving the food by organic acid and alcohols, impart desirable flavor, texture, and reduce toxicity and decrease the cooking time of the food. Fermentation occurs within the gastrointestinal tracts of all ruminant animals, including humans. So we all know that even in our body, in our intestinal, the gut, fermentation happens. We have a lot of microorganisms in our gut too. So let us see the definition of fermentation. Fermentation can be defined as the chemical modification of organic compounds or raw materials or agricultural commodities with the aid of enzymes produced by microorganisms. With respect to foods, it is the transformation of physical structure and chemical constituents like carbohydrate, protein, fats, and nucleic acids by microorganisms and their enzymes under aerobic or anaerobic conditions. So in fermentation we can see both conditions. So initially there will be plenty of oxygen, so later it will be converted to anaerobic condition. Okay, so examples for some fermented food products are wine from grapes, cider from apples, that is apple cider vinegar we usually use, then beer from malted barley, whiskey, gin, rum, pickles, curds, idli or dosa, then bread and butter naan, that is naan, cheese, and lactic acid beverages. During fermentation, the food product undergoes many changes physically, chemically, as well as nutritionally. Fermentation is a natural way to preserve variety of food products. Fermentation enhances the organoleptic attributes of foods like texture, flavor, aroma, etc. So isn't it right, correct? So we have many fermented food products, like if you take yogurt, there are a number of yogurt products, flavored yogurts available in the market. So the texture, taste, flavor, aroma, everything is because of the fermentation. Preservation of food by fermentation improves the nutritional composition of the food product, hence making it a better food product for consumption. Fermentation is a cost-effective method for food preservation as it needs smaller amount of starter culture which can ferment large amount of food at one go. So even at our kitchen level we do the fermentation. It doesn't require much of um, sophisticated instruments or equipments and all. Fermented food products boost our immune system by preventing pathogenic microbial infection in our body. Thus, it strengthens our overall well-being and health. Let us see the history of fermentation. Fermentation has been used by man for thousands of years. So initially if we see in Iraq around 6000 BC, cheese production was first developed. Okay, so it is so long we can see, so long back we can see the cheese production in Iraq where it was started first. Later in Egyptians found the use of yeast for leavened bread making and wine production. This was followed by discovery of beer from fermented barley by Sumerians. Later came the discovery of antibiotic from moldy soya bean which was used by Chinese in 500 BC. Then first distillery of whiskey was established in Ireland. In 1500 AD, yogurt and sauerkraut fermentation was also discovered. After all these discoveries, niacin as a preservative was discovered, which is an inhibitory protein substance isolated during cheese making in England. So we'll see about this niacin in the chemical preservatives section again. So one major scientist who contributed a lot for this fermentation microbiology is, we know that it is Louis Pasteur. So pasteurization, which is a process that involves heating and rapid cooling treatment, was developed by Louis Pasteur in 1861. The French chemist Louis Pasteur was the first zymologist when in 1857 he connected yeast to fermentation. So he is the one who found the connection between the microorganism yeast to the fermentation. French chemist Louis Pasteur originally defined fermentation as respiration without air, that is respiration in the absence of oxygen or in the anaerobic condition. Louis Pasteur was one of the pioneer researchers on food preservation who proclaimed that the microorganisms that are developed from tiny inoculums are not spontaneously generated. Okay, it is either naturally present in the food itself or it is added later. That was found by Louis Pasteur. Let us see the changes happening during fermentation. Both harmful and useful microorganisms grow in the environmental and ecological conditions in food commodities, food preparations and processed foods and other systems, and produce enzymes, metabolites and toxins depending on nutritional and physical factors. So we know that we have useful microorganisms as well as harmful microorganisms also. Both also grow. So we want the useful one to be focused now. So these factors are the factors which influence the growth of microorganisms are water activity, pH, temperature, chemical constituents and the buffering compounds of the food. So we have two types of fermentation. Okay, so one is naturally happening, another is controlled, where we control the process of fermentation. Okay, in natural fermentation, so we don't add any microorganisms or the inoculum or starter culture, and in controlled fermentation, so we add the starter culture and we control to what extent the microorganisms ferment the food. Okay, microorganisms bring about many chemical and structural changes in organic constituents of foods and thus check the growth of pathogenic microorganisms. The lactic acid bacteria convert available carbohydrate to lactic acid, where by lowering the pH and changing the conditions suitable for the growth of yeast. So where do we see the lactic acid bacteria mostly in our kitchen? So it is basically in the curd, right? Sometimes propionic acid is also produced, which acts as a preservative. So this propionic acid is also used as chemical preservative, that we will see in the next section. Okay, so this is also produced during fermentation sometimes. They also produce flavor compounds like diacetyl, acetaldehyde and acetoin, which gives pleasant flavor to the fermented food products. The acids developed by the organisms during the processing of food contribute to the flavor of the final product and also acts as preservatives, preventing the growth of undesirable pathogenic and spoilage microorganisms. So this is the major change which we can observe in fermentation, that is the acid produced in the fermentation process. So this acid produced will give you good flavor, as well as it is the main preservative in the fermented food products. Some typical examples of fermentation byproducts are ethanol, lactic acid and hydrogen gas. Several other exotic compounds can be produced by fermentation, that includes butyric acid and acetone also. Fermentation process. So let us see the process of fermentation. Fermentation essentially takes place in anaerobic conditions when there is no oxidative phosphorylation to maintain the production of ATP, that is the energy bank adenosine triphosphate by glycolysis. During fermentation, pyruvate is metabolized to diverse compounds. So we can see three kinds of fermentation happening usually. First is homolactic fermentation, where you can see only the lactic acid production from the pyruvate. The second type is heterolactic fermentation, so where you can see the production of lactic acid as well as other acids and alcohol production during the fermentation. And the third type is alcoholic fermentation, where pyruvate is converted to ethanol and carbon dioxide majorly. Yeah, so the two types of fermentation which we commercially uses ethanol fermentation as well as lactic acid fermentation. So let us see about ethanol fermentation. Ethanol fermentation is the process in which glucose is converted to ethanol and carbon dioxide. Ethanol fermentation is also known as alcoholic fermentation, that converts sugars such as glucose, fructose and sucrose into energy, ethanol and carbon dioxide as the byproducts. Ethanol fermentation or yeast fermentation have wide practical applications in the production of alcoholic beverages, then ethanol, then cheese and bread. This process is mainly responsible for the existence of any sort of alcohol. Yeah, the second type is lactic acid bacteria fermentation, that is lab fermentation. It is done by using natural microflora or lactic acid bacteria cultures, which is employed throughout the world in conjunction with the chemical preservation using salt and acid to preserve various foods such as milk, cereals, meat, and fruits and vegetables. The lactic acid bacteria tolerate high salt concentration, which give them an advantage over other less salt tolerant species and allows the lab to produce acid that inhibits the growth of undesirable microorganisms. So what happens in lactic acid bacteria fermentation? So the pH is being reduced due to the production of acid, right? But still the microorganisms, as they are resistant to high acid and salt concentration, they can survive at lower pH also. Lactic acid fermentation enhances the organoleptic and nutritional quality to the fermented fruits and vegetables and retains the nutrients and colored pigments in the food. Two of the most common application of lactic acid fermentation in the food industry is the yogurt and sauerkraut. The sauerkraut is made from the cabbage fermentation and yogurt is made from milk fermentation. Let us see the main uses of fermentation. So fermentation helps in the improvement in the profile of flavors, aroma, texture of the fermented foods. So fermentation also helps in preservation through lactic acid, alcohol, acetic acid and alkaline fermentations. So fermentation also helps in the enrichment with essential amino acids, essential fatty acids and vitamins. That is, nutritionally the quality of the fermented foods will be higher when compared to others. So fermentation also helps in detoxification of certain toxic constituents during food fermentation. So some anti-nutritional factors are also destroyed during fermentation. So that's all about the process of fermentation. Let us see about some fermented food products available in the market. So the first one, which is being used widely nowadays, that is cheese. Cheese is a milk-based product obtained from casein, that is the milk protein coagulation. Milk contains lactose sugar, which is subjected to fermentation to produce lactic acid by addition and action of bacterial culture. So the bacterial culture used for cheese production mainly there are a variety of bacteria. So two of the common ones are Streptococcus cremoris and Streptococcus lactis. So you must be knowing that there are variety of cheeses due to the action of varieties of microorganisms. So each microorganism acts on the cheese. It gives different flavors as well as different colors. Based on that, we have soft cheese, hard cheese, semi-hard cheese, like that. Varieties of cheese are there and the microorganisms are vast. Milk is then left with coagulated casein proteins. Then the separation of casein from whey is done. So what are we doing? First we are fermenting the milk. So lactose is fermented and the milk is left with the coagulated protein, right? That coagulated protein we are separating from the whey, okay, the liquid part of the milk. After the cheese is left for ripening by microbial action to obtain cheese followed by its proper packing. So once the casein protein is separated, it is allowed for the microbial action. So this is called cheese maturation. Okay, so the longer we keep for maturing, the cheese maturing, the higher will be the flavor of the cheese. Shelf life of cheese is increased as a result of fermentation, and it can stay unspoiled if it is kept in a proper way up to several weeks to several months. Next is wine. Wine is an alcoholic beverage or liquid which is significant for both industries as well as for commercial purpose. Wine making starts with fruit selection. So the common wine available is grape wine, right? We have variety of wines now, but the common one is grape wine. So grapes, which has important acids, esters and other compounds which are required to make wine naturally. Wine making starts with fruit selection, that is grapes, which has important acids. Then squashing of the grapes are done, that is mashing of grapes to obtain the pulp. Then it's the turn for fermentation, which occurs naturally within some hours, that is 6 to 12 hours, by addition and action of yeast. Fermentation is continued till dry wine is obtained by the transformation of sugar present in grapes to alcohol. Fermentation occurs for longer period of time, so it is stopped before whole of the sugar is converted to alcohol to get sweet wine. So if you want sweeter wine, you have to stop the fermentation in between in order to save some sugar which is there in the wine. Lastly, classification, then bottling and aging process completes produced the final product of the wine. The production of the final product of the wine. Fermentation of wine results in its longer shelf life, and if the wine is kept in a proper manner in appropriate conditions, then it can stay unspoiled up to some days to years, depending on the type of wine. So the major microorganisms involved in wine production is the Saccharomyces species. Okay. Yeah. Next product is soy sauce. The soy sauce or the soya sauce is a very popular food ingredient and is widely used in Chinese, Japanese and Indo-Chinese cuisines. It is a dark colored fermented liquid which is salty in taste and is a product of soy beans or the soya beans. Soya sauce, due to fermentation preservation, can be stored for about 6 months or more if kept in conditions, in proper conditions and temperature. To obtain good quality of soya sauce, firstly, soya beans are cooked for about 4 to 5 hours and then left for cooling. After this, equal quantity of roasted and ground wheat is added. Then the mixture is seeded with Aspergillus oryzae for initial fermentation process followed by addition of salt. So what do we do first? Equal quantity of roasted and ground wheat is added to the soya beans. Okay, the cooked soya beans. So then it is seeded with which microorganisms? It is Aspergillus oryzae. So you can remember this microorganisms, which is important. This whole mixture is left for maturation for about 6 months to 3 years when further fermentation occurs. The product is then finally strained and packed. So you can see the picture of the fermented soya sauce here. It is dark in color. Yeah. Next is idli. So idli is a very common breakfast dish in South India. Idli is prepared by the appropriate mixture of soaked rice and black gram, and then it is ground. Overnight fermentation of idli batter allows the rapid growth of the naturally occurring microorganisms, which are Leuconostoc mesenteroids and Streptococcus thermophilus, in grains or legumes or in utensils. So what is the source of these microorganisms? It is basically from the black gram, okay, the urad dal or the black gram, or from our hands when we mix the idli batter with our hands. The microorganisms from our hands also help in the fermentation, or the microorganisms present in the utensils. These microorganisms divide and produce lactic acid and carbon dioxide that makes the batter anaerobic and leaven the product. Bubbles of carbon dioxide form a froth on top of the fermentation mixture. Yeah. Next is yogurt and kimchi. To turn milk into yogurt, bacteria ferments the milk, turning the lactose sugar in the milk into lactic acid. Some of the curd or yolk bacteria are Streptococcus thermophilus, then lactic acid bulgaricus, lactic acid acidophilus, lactic acid casei, lactic acid rhamnosus, Bifidobacterium animalis, and Bifidobacterium bifidum. So these are the yogurt or the curd microorganisms. The lactic acid is what causes the milk as it ferments to thicken and taste tart. Because the bacteria have partially broken down the milk already, it is thought to make yogurt easier for us to digest. So the protein and other compounds in the yogurt is already partially digested by bacteria, so it will be easy for us to digest. The next product is kimchi. So kimchi is basically a Korean dish. It is a staple food in Korean cuisine. And this kimchi is a traditional side dish made from salted and fermented vegetables, most commonly the cabbage, napa cabbage, and Korean radishes, with a variety of seasonings including chili powder, scallions, garlic, ginger, and geotgal. So geotgal is kind of spice used in the Korea. So what is the major ingredients which we use? The salted cabbage and Korean radish. Along with that, seasonings we use are chili powder, scallions, garlic, ginger, and geotgal. Yeah. Some other examples for fermented bacteria using lactic acid bacterial fermentation are pickle and sauerkraut. So pickle is a product prepared by lactic acid bacteria fermentation of sugar present in pieces of fruits and vegetables. The prepared product is rich in lactic acid, and only the beneficial bacteria that can tolerate lactic acid pH survive in pickle. So in pickle, fermentation is one of the preservative. Along with that, we have a lot of other preservatives like oil, and also in some areas they use mustard as the preservative. So in pickles there are multiple preservatives are used. Next one is sauerkraut. So it is basically a finely cut cabbage that has been fermented by various lactic acid bacteria. Sauerkraut usually has a long shelf life and a distinctive sour flavor. Okay, that's fermentation as a preservative. Next is preservation using chemical preservatives. Food preservation is a method of preparing food so that it can be stored for future use. We all know that. Because most foods remain edible for only a brief period of time, people since the earliest ages have experimented with methods for successful food preservation. So, such preservations are done in two methods. One is physical method and chemical method. So the physical methods are the ones which we have studied previously, drying and all, right? So physical methods of preservation rely on killing the microorganisms present, or at least stopping their growth for long enough to allow the food to be safely consumed. The physical methods, if we take some examples, it is canning, freezing, drying, gamma irradiation, then ultraviolet or high intensity white light preservation, then ultra high pressure and filtration, ultra high pressure filtration method. So this is about the physical methods of preservation. Then comes the chemical food preservatives. So these are the substances which under certain conditions either delay the growth of microorganisms without necessarily destroying them, or they kill them. These are added in very low quantities and which do not alter the organoleptic and physiochemical properties of the food at or only very little. So the chemical preservatives, when it is added, it will not change the organoleptic property, or it will enhance the organoleptic properties of the food. Along with that, it will be in a safer level, so it will be safe for the consumption too. Yeah. The Food, Drug and Cosmetic Act permit for the use of chemical preservatives in foods if the chemical is, so the following are the conditions to permit the chemical preservatives to be used in the food. Okay. Generally recognized, if the chemicals are generally recognized as safe, that is GRAS, for such use, or if a food additive is covered by food additive regulations prescribing conditions of safe use, or these chemicals are not used in such a way as to conceal damage or inferiority, or to make the food appear better or greater value than it is. So when these chemical preservatives are added, it should not be a fraudulent act where the consumers are fooled by changing its appearance or taste or any other characteristics, external characteristics of the food. So the next thing is the chemical preservatives should properly be declared on the label of the food in which it is used. So it should be food grade chemical preservatives, then it should perform its intended function, it should be used in accordance with good manufacturing practices, and where applicable according with existing food additives regulations. So in order to select some chemical preservatives, it should be within the given regulations, and also it shouldn't be fraudulently used, or it has to be mentioned in the label of the food product. According to rules, a food manufacturer must get approval from government regulatory authorities before using a new preservative or before using a previously approved preservative in a new way or in a different amount. In its petition for approval, the manufacturer must demonstrate that the preservative is safe for consumers considering the following points. So, the points to be considered before approving one chemical preservatives are the probable amount of the preservative that will be consumed with the food product, or the amount of any substance formed in or on the food resulting from the use of the food preservative. And the next point to be considered is the cumulative effect of the preservative in the diet. Then the potential toxicity, including cancer causing toxicity of the preservative when ingested by humans or animals. Considering all these points, it will be allowed to be used as preservative or not. Yeah. A preservative may not be used to deceive a consumer by changing the food to make it appear other than it is. So let us understand more about this. If we take an example, preservatives that contain sulfites are prohibited on meats because they restore the red color, giving meat a false appearance of freshness. So whenever you use some preservative, even though it is safe for consumption, if it does not cause any harmful ill effect to our health, it should not be used if that chemical preservative is giving a false appearance of any food product. So in this case we took the example of meat, right? So if you add sulfites, it will restore the red color, so that will make the food appear brighter, right? This is a false appearance, so that should not be used as preservative. The food additive regulations require the preservative to be of food grade and be prepared and handled as a food ingredient. So these preservatives should be used as a food ingredient. The quantity added to food must not exceed the amount needed to achieve the manufacturer's intended effect. So the quantity given by the regulations should be maintained the same. In order to achieve better preservation, the manufacturer shouldn't be increasing the quantity above the limit. Let us see the classification of preservatives. There are different types of classifications. So let us see one type of classification now. First type is antimicrobials that inhibit growth of bacterial yeast and or molds. Then antioxidants that slow air oxidation of fats and lipids, which leads to rancidity. The next type is anti-enzymatic that blocks the natural ripening and enzymatic processes that continue to occur in foodstuff after harvesting also. Then preservatives from natural products, then traditional preservatives. So what are the five types? Antimicrobials, antioxidants, anti-enzymatics, then natural preservatives and traditional preservatives. So one more type of classification we have, that is class 1 preservatives and class 2 preservatives. This is a very simple classification in which you can uh, come to know that which is natural and also available, easy to use in the kitchen, and which is industrial level. So in class 1 preservative, it includes sugar, salt, spices, vinegar, alcohol. So these things are relatively safe to use. So when it is added to food, it does not have a proper limit for consumption, even it gives better taste. The second type is class 2 preservatives. This includes benzoic acid, sulfur dioxide, nitrites and nitrates, and a variety of neutralizers, firming agents and bleaching agents. These are referred to as class 2 preservatives, and these are also considered to be relatively safe to humans, but within the permissible doses prescribed by the food regulatory bodies of the country, because higher concentration of these class 2 preservatives can be a health hazard. Yeah. So let us see different types of preservatives based on the first type of classification we saw. Okay, so all these five types comes under the second type also, as it has more detailed classification. We'll look according to the first classification. So the first type is antioxidant preservatives. As antioxidants, they keep foods from becoming rancid, browning, or developing black spots. Rancid foods may not make someone sick, but they can smell and taste bad. That's why it is important to avoid rancidity in the food. Antioxidants suppress the reaction that occurs when foods combine with oxygen in the presence of light, heat, and some metals. That is, antioxidants avoid the oxidation of food. Antioxidants also minimize the damage to some essential amino acids, the building blocks of proteins, and the loss of some vitamins. Antioxidant preservatives such as butylated hydroxytoluene, butylated or butylated hydroxyanizole, third butyl hydroquinone, then propyl gallate. These antioxidant preservatives stop the chemical breakdown of food that happens in the presence of oxygen. Unsaturated fatty acids in oils and lipids are particularly susceptible to auto-oxidation. In this process, a free radical initiates peroxide formation at fatty acid double bonds. The chain reaction propagates to other double bonds, and aldehyde, ketone, and acid termination products, eventually building up to create the rancid of flavor characteristic of oils and fats gone bad. So this is how rancidity occurs. Rancidity occurs very fast in unsaturated fatty acids because they have the double bonds which are susceptible to such oxidation. Antioxidant preservatives pop up the free radicals that help initiate and propagate these reactions. Okay, that's all about antioxidants. Next is about the enzymes, the preservative enzymes. These are preservatives that target enzymes in the food itself that continue to metabolize after harvest. The enzyme phenolase, for example, goes to work as soon as the apple or potato is cut. It browns the exposed surface area of the food. Acids such as citric acid, ascorbic acid inhibits phenolase by making the pH chemically uncomfortably low for the enzyme. So what does these enzymes do? So these these acids basically reduce the pH, which is not favorable for the enzyme activity. Metal chelating agents such as EDTA, ethylene diamine tetraacetic acid, can remove the metal cofactor that many enzymes need. So enzymes need cofactors for their activity. So in order to have their reactions faster, they require these cofactors. So these cofactors are blocked by EDTA in the process. Chelators also make it difficult for bacteria and fungal enzymes to carry on the deterioration in the food. So next is preservatives from natural products. Some of the newest antimicrobials have been found in microorganisms themselves, as they form their own chemical defenses when competing with each other for space and nutrients. So these natural antimicrobials are produced by microorganisms while defending themselves from other microorganisms. For example, niacin and natamycin, the cheese preservatives called bacteriocins, are harvested from microorganisms themselves. Niacin is used to inhibit the outgrowth of Clostridium botulinum spores and toxin formation in pasteurized processed cheese spreads with fruits, vegetables or meats at levels not exceeding good manufacturing practices. Current good manufacturing practice in the case is the quality of the ingredient that delivers a maximum of 250 parts per millimeter of niacin in the finished product. Niacin is considered effective at controlling a wide range of gram-positive organisms including Listeria enterococcus, Bacillus sporothermodurans and Clostridium species. When they are used alone, it is not effective on gram-negative bacteria like E. coli, yeast and molds. However, researchers suggest that it may be used against some gram-negative bacteria when used in conjunction with other preservatives. So niacin is one of the examples which are used as a preservatives in cheese preservation, right? So this niacin is extracted from some microorganisms, so which produce this niacin in order to defend from Clostridium species, Listeria species and Bacillus species. Next is traditional chemical food preservatives. Traditional chemical food preservatives are used in fruits and vegetable processing technologies. So the best examples are salt and sugar. Common salt used in brine vegetables, they don't have any limit for their use. Sugars like sucrose, glucose, fructose and syrups, they are also used as foods preserved by high sugar concentrations such as jellies, preserves, syrups, juice concentrations, etc. So these salt and sugar acts by interacting with other ingredients and by reducing the water activity of the food. So there is no limit for the use of this salt and sugar. According to the taste, it can be used. Yeah. So next is about the commonly used antimicrobial chemical food preservatives. So let us see one by one. So we have the sorbates, mainly the sorbic acid and potassium sorbate. The acceptable daily limit is 25 milligrams per kg body weight. The commonly used levels is 0.05 to 0.2%. So it is typically used in fruits, vegetables, pickled products, jams and jellies. Then comes the benzoates, that is benzoic acid and sodium benzoate. So it can be taken as 5 milligrams per kg body weight, and the commonly used level is 0.03 to 0.2 percentage, which are used mainly in vegetable pickles, jams, jellies, semi-processed products. Then comes the propionates, that is propionic acid and sodium propionate, which can be used up to 10 mg per kg body weight to the level of 0.1 to 0.3% in foods like in foods like bakery goods, cheese spread, fruits, vegetables, etc. Then comes the parabens, methyl paraben, ethyl paraben and propyl paraben. It can be used up to 10 mg per kg body weight to the levels of 0.05 to 0.1% in foods like bakery goods, fruit products, pickles, sauces, etc. Then is the in some acids like lactic acid, citric acid and acetic acid. These acids have no limits. They only to the acceptable taste level it can be used. And lactic acid is used in fermented meat, dairy and vegetable products, sauces and dressings, drinks, etc. Citric acid is used in fruit juices, jams and other sugar preserves. Then acetic acid is used in pickles and sauces, chutneys, etc. Then sodium nitrate is allowed to take up to 0.2 mg per to the level of 0.01 to 0.02 percentage in food products like meat products. Next comes the sulfur dioxide, which can be taken daily up to 0.7 milligram per kg body weight to the level of 0.005 to 0.2% in foods like fruit juices, dried fruits and vegetables, semi-processed food products. So these are the commonly used antimicrobial chemical food preservatives. So that's all about the fermentation and chemical preservation in the preservation of foods. For any query, you can contact the number on the screen. Keep studying with iiocs.com. Thank you. Goodbye.